Domestic dogs exhibit tremendous phenotypic diversity, including a greater variation in body size than any other terrestrial mammal. This diversity is the result of years of interbreeding between wolves and other descendants of dogs. However, in recent years, human selection has played a huge role in the breeds of dogs you see today. The phenotypic diversity of the dog, its unique breed structure, and growing genetic resources present a powerful system for genetic dissection of traits with complex inheritance. Over the past few years, many genetic variants and quantitative trait loci, or QTL, have been identified, which influence certain traits such as skeletal size, coat colour, and leg length, which is used to differentiate between breeds. Dogs were selected for this study as they offer a unique system of genes controlling morphology. DNA from 915 dogs from 80 domestic breeds, also a select number of feral dogs, were tested, using over 60,000 points of variation in the dataset analysed using methods to find loci regulating body size, head shape, leg length, ear position, and a host of other traits. Due to such strong selection by breeders for dogs to have a preferred appearance, there is a solid footprint of selection in regions of the genome that are needed for controlling these breed-defining traits. The analysis identified regions of the genome and loci that are important for controlling body size and shape. As shown in Figure 1, it is found that while LD extends over 1 megabase within every breed surveyed, across all dogs combined it decays extremely rapidly, consistent with previous studies. This suggests few IVD segments are shared across multiple breeds, and those that are shared are quite small, which is seen in Figure 1a. Homozygous runs are also longer and more numerous in bred dogs than village dogs or wolves as viewed in Figure 1b, with individuals from nearly every breed exhibiting 10 to 50 runs of homozygosity and greater than 10 megabase. Interestingly, Jack Russell Terriers are an extreme outlier, with fewer runs of homozygosity and overall higher levels of diversity than any other dog breeds. In contrast to human populations, where patterns of autozygosity in populations are not generally correlated with haplotype diversity, purebred dogs show a very strong negative correlation between autozygosity and haplotype diversity, as shown in Figure 1c. One notable exception is Basenjis, which show the high haplotype diversity and high autozygosity, suggesting a recent population bottleneck post-breed formation has induced higher levels of inbreeding than expected. This is consistent with the breed history of Basenjis in the United States, which are believed to descend from a smaller founder population. The results demonstrate the efficiency of mapping multiple traits in the domestic dog using a database of genotyped individuals and highlight the important role human-directed selection has played in altering the genetic architecture of key traits in this important species.